goal of protein digestion is breaking proteins all the way down to the individual amino acids that they are made of. By doing this, as you recall, their original function will be lost, which is not a problem because we eat protein only to get the pieces, the amino acids that we will then use to build our own proteins. To break down proteins, we do two things: denaturation and enzymatic hydrolysis. Denaturation allows us to unfold proteins so that they can then be more easily accessed by our digestive hydrolytic enzymes, which will start breaking the peptide bonds to create smaller and smaller protein fragments until all the individual amino acids have been separated. These amino acids will then be absorbed and used by the cells in our body to build the proteins that we need. We could consider cooking to be the first step of protein digestion. As you recall, exposure to heat causes protein denaturation, thus making it easier to then complete protein digestion. By softening tissues, cooking also increases the bioavailability of proteins from many foods. Not much happens in our mouth, and the first real digestive step is in the stomach. Here, the strongly acidic environment completes protein denaturation and unfolding. The proteolytic enzyme pepsin starts hydrolyzing proteins. It is not a thorough job, it just starts cutting protein chains here and there until smaller protein fragments will be created from longer chains. The real protein breakdown takes place in the small intestine. Here we have a family of proteolytic enzymes from the pancreas, led by trypsin, which make these chains smaller and smaller until they are small peptides of just a few amino acids. These small peptides will then reach the brush border, the walls of our intestine, where we have the intestinal absorptive cells, the enterocytes. Here, another family of proteolytic enzymes will complete protein hydrolysis to the individual amino acids, and this can happen on the brush border right before absorption, but also inside our intestinal absorptive cells. Indeed, we do have carriers to absorb small peptides, 2 or 3 amino acids long, into our enterocytes, and once they are inside, the enzymes will complete their breakdown into the individual amino acids before they are released in the bloodstream. Notice that if we were to eat single hydrolyzed amino acids, our digestion would be easier, but our absorption would be way less efficient because we wouldn't be able to use these carriers for di and tripeptides on our brush border. For this reason, supplements of individual amino acids are better absorbed if they're actually made of a mix of single amino acids and small peptides. Either way, once they are inside the intestinal absorptive cells, our proteins have been broken all the way down to single individual amino acids. These amino acids can now go through the intestinal wall into the bloodstream. Amino acids are water soluble, so they can go directly in the bloodstream by themselves. From the small intestine, they take the portal vein and go to the liver. The liver will sort them out depending on what the body needs to do with these amino acids that were just absorbed. They can be used for protein synthesis or for energy production, and if we don't immediately need them for protein synthesis or energy production, then, since we cannot store them, we will convert them to either fat or glucose that can be stored. We will see all these different uses of proteins later on, but for now let's just remember that it's the liver that does this sorting activity and decides where the amino acids go and what they should be used for. There are actually 3 amino acids, the branched chain amino acids, leucine, isoleucine, and valine, that skip this liver step and go straight to the muscle, which will use them for energy production. We will also return on this later. There are some exceptions to the general rule that with digestion we completely break protein down, lose their function, and only absorb the individual amino acids. Sometimes, very small proteins or protein fragments can leak through the intestine in different ways, and this especially happens in infants younger than 5 months. Their intestine is a little bit more permeable, and so these small peptides can go through without being completely broken down. This has 2 main consequences. The first one is allergies predisposition, because when we have a whole protein or a piece of protein inside our body coming directly from outside, it may be recognized as something foreign and activate some kind of immune response, which is the basis for possibly developing an allergy. For this reason, it is recommended that infants until 5 or 6 months do not get those foods that contain proteins frequently associated with allergies, such as nuts, fish, or strawberries. The other consequence is that if this protein or protein fragment has a biological function, this function will be preserved. And so now we have inside our body a protein that has a function, but that we didn't build ourselves. For some small peptides, this can happen in adults as well. For example, there are some casomorphins in milk that have opiate-like activity and that are sometimes absorbed intact. Some people claim that they cannot sleep without drinking milk, and some researchers hypothesize that this happens because there are these little peptides that have this calming activity.